Hi students, I'm going to be finishing up the Chapter 11 lecture on muscle tissue physiology. So previously we discovered how muscle contraction actually works, so the physiological steps that occur from the time a nervous impulse is sent down the axon of a neuron to the neuromuscular junction, and we looked at all of the changes that occur on the sarcolemma and then within the cell itself. Now I'm going to get into more behavior of whole muscles rather than just muscle cells. The first topic that I want to talk about is the length-tension relationship. This is simply stated as the tension that's generated by a muscle, and therefore the force of a contraction is going to depend on how stretched or contracted the muscle is at the outset. So this is a fairly intuitive concept, I think. If a muscle is already fairly stretched out, there's only so much more length that can be generated. Whereas if a muscle is already contracted, it's already generating its maximum or close to maximum amount of tension. So it cannot generate much more. So if a muscle is too contracted, that means that its thick filaments are already very close to the Z discs, which are those boundaries of an individual sarcomere. The contraction is going to be brief and weak. So you can only contract a muscle so much. If you are trying to contract a muscle more, there are physiological limitations to that, and that just has to do with the relationship of those filaments to the Z discs. If a muscle is too stretched, that means there's too little overlap between thick and thin filaments. So the myosin heads are not going to be able to maximally grip the binding sites on actin. There's just not going to be enough initial overlap there for myosin to act on actin. So that is the length-tension relationship. We can look at theoretical calculations that are based on this principle. So on the x-axis here we have sarcomere length in microns or micrometers before stimulation occurs. And then on the y-axis we have the relative tension that's generated once stimulation occurs. So if we look at optimum resting length, this would be the length between one Z-disc and another, so the length of an individual sarcomere optimally before contraction is going to be 2 to 2.25 microns. That means that there is enough overlap between myosin and actin that you're going to have plenty of contact between those myosin heads and actin binding sites, such that you can contract maximally from that point. If a muscle is already overly contracted, then there's only so much overlap that can occur from that point. And as you see here, we've already basically reached the maximum amount that that muscle can contract. So further stimulation is not going to be very productive in terms of generating tension. And then if a sarcomere is overly stretched, you see that there's just very little opportunity for the myosin heads to actually act on those binding sites of the thin filament on actin. So these calculations are based on theoretical calculations. In real life situations, you're not going to see this amount of stretch or this amount of contraction typically. There's a few terms that I would like you to be familiar with as well. Chapter 11 is a fairly in-depth chapter, and if you're going into physical therapy or exercise and sports science, I highly recommend taking the time to become familiar with the parts of the Saladin textbook that we just don't have time to cover in this class. But there are a few terms that you may already be familiar with that are going to be helpful going forward. So muscle tone is a partially contracted state of muscle tissues that the central nervous system maintains. This is particularly important for muscles that relate to posture, so muscles that help us in maintaining sitting upright or standing, for example. So, a lot of the examples that I've given in class have been looking at the movements that occur at a joint, flexion and extension. And, if I want to lift this particular weight, how does that relationship between the nerve and the muscle tissue work to actually do that work? But it is important to keep in mind that many of our muscles are just maintained in a partially contracted state. And that is going to be particularly important for those muscles related to posture. Another term that you are probably familiar with is muscle fatigue, which is just a progressive weakness and loss of contractility that results from prolonged use of muscles. The cause of fatigue can vary depending on, the exercise type, or it can be from overuse, for example. And then slow twitch and fast twitch fibers. Slow twitch fibers are muscle fibers that are adapted to endurance. They're also known as oxidative fibers, and they generally have a lot of resistance to fatigue because they rely on aerobic respiration, which supplies the tissue with enough ATP to undergo continuous contraction. So a slow twitch fiber is going to be indicative of a muscle fiber that's adapted to being used a lot. Whereas a fast twitch fiber is going to be a muscle fiber that's adapted for quick responses and bursts of power. So for example, like the gastrocnemius in the leg. Fast twitch fibers generally use anaerobic sources of energy. So rather than aerobic respiration, fast twitch fibers are going to rely on glycolysis, for example, which is a form of generating ATP from sources of fuel that does not rely on the presence of oxygen. So there are some physical differences that are apparent between these two types of fibers. The fast twitch fibers have less myoglobin because they're not as reliant on oxygen, and myoglobin delivers oxygen to muscle tissue. So as a result of this less myoglobin in these fibers, it is a paler tissue. And you can see this difference if you look at the raw or cooked meat of a chicken. The chicken breast, for example, is going to be comprised largely of fast twitch fibers. Chickens are able to fly in short bursts, and the pectoralis muscles of the breast are adapted to using very little oxygen, so they're relying on glycolysis primarily. So those fibers are going to be very pale in color. They're basically white, whereas the legs of the chicken are going to be adapted for endurance, for walking, for example. So they are going to be more heavily populated with myoglobin, and the fibers themselves are going to be darker in color as a result of that myoglobin. Okay, so there's a couple of terms related to types of contraction of muscle tissue. These are isometric and isotonic contractions. With an isometric muscle contraction, what happens is the muscle produces internal tension, so tension is being generated at the sarcomere level, but the external resistance is causing that muscle to stay the same length. So there's too much resistance to actually see a change in length, at least for a very brief period of time. So this can be a prelude to movement when the tension of, say, picking up a heavy weight is going to be initially absorbed by the elastic filament components of muscle. So if you go to lift a heavy weight, for example, there may be just a split second where your muscle has not, there hasn't been enough summation in the muscle fibers to actually generate enough contractile force to actually lift that. So some of the initial tension is just going to be absorbed by those elastic filaments. and you don't actually have a change in length, so you're not moving the muscle, or you're not moving the weight immediately. So again, it can be a prelude to movement, but it's also important in postural muscle function, as well as antagonistic muscle joint stabilization. So muscles that act as antagonists to prime movers, for example, that help to stabilize a joint. They will often utilize this isometric muscle contraction. So we see this here if we're initially picking up a weight, especially if we haven't lifted a weight in a while, might not expect it to be quite as heavy. The muscle is going to develop tension, but it's not going to shorten immediately. So this would be a prelude to movement. With isotonic muscle contraction, the muscle is going to change in length, but it's not going to have any change in tension. So there are two different categories to this isotonic muscle contraction. Concentric contraction is when the muscle shortens as it maintains tension. So, for example, lifting a weight. You are shortening the muscle. So biceps brachii, for example, all of those sarcomeres within that muscle are going to be shortening. Not all, but largely they are going to be trending towards shortening. And the tension is going to remain constant. So you don't have a jerky movement. You have this smooth movement of the weight as you're maintaining tension, but the muscle is shortening. And then an eccentric contraction is when the muscle lengthens as it maintains tension. So this would be, an example of this would be when you're lowering a weight. So biceps brachii, again, you are having to maintain tension to avoid just dropping that weight, but the sarcomeres are largely lengthening. Okay, let's see. So cardiac muscle, I'm going to be talking about cardiac and smooth muscle just briefly to kind of compare and contrast those to what we've been talking about in relation to skeletal muscle. Cardiac muscle, of course, is only found in the heart. They tend to be kind of branched, irregularly shaped cells with a central nucleus. They do have striations, and they have what are known as intercalated discs, which we saw in our histology slides as just kind of irregular horizontal lines dispersed throughout the tissue. Some of the characteristics that make cardiac muscle unique is, of course, it must contract with a regular rhythm. So we have this pattern of filling the chambers of the heart and then expelling blood from the chambers of the heart. Something that we see with cardiac muscle is that, of course, it must be functioning constantly. It has to function both in sleep and wakefulness. It has to be highly resistant to fatigue. The cells must contract in unison, so there has to be some basically cooperative nature to contraction and relaxation. And then contraction also has to last long enough to expel blood from the chambers. So with cardiac muscle cells, we actually have what are known as pacemaker cells, which basically set the pace of blood expulsion and filling of the chambers. And due to the fact that we have these pacemaker cells and the fact that the heart doesn't actually have to have nervous system stimulation in order to contract, we also have this property known as autorhythmicity. So the heart is known as being autorhythmic because auto means self. So it basically sets the pace of this very predictable rhythm of contraction and relaxation by itself without having to have nervous system stimulation. That's not to say that there are not connections between the nervous system and the heart, because of course there are, but the myocytes themselves do not have to have that stimulation from a nerve like we saw with skeletal muscle tissue in order to contract. And then these intercalated discs, basically, there are a number of proteins within these structures that help, basically, it helps the signal to spread from one cell to another. So that helps with basically contracting in unison. And there are special types of linkage proteins that help to anchor one cell to another because these cells are under a lot of stress. And the special proteins within those intercalated discs help keep that tissue stable and prevent it from tearing. So when you get to AMP2, if you are going into the second semester of our anatomy and physiology class, you will be learning more about the unique properties of cardiac muscle, but just kind of as a sneak peek of some of the topics that you'll be talking about and some of the properties that you want to be thinking about, like how does the cardiac muscle accomplish these things? There's always going to be this interrelationship between structure and function. So oftentimes, looking at the structure of individual cells and then the heart itself as an organ is going to help you understand those differences. Okay, so with smooth muscle, now think back, where do you find smooth muscle? Smooth muscle is going to be found, for example, in blood vessels, the walls of blood vessels, the walls of digestive system organs. It's going to be in piloerector muscles within the dermis to control erection of hairs in the case of goosebumps, for example. It's even going to be in the eye, the iris of the eye, to control how dilated the pupil is. So smooth muscle is found in all of these places where we need to create movement in the body, but it's not under voluntary control. So it is involuntary. It is generally part of this system between the autonomic nervous system and smooth muscle tissue. So your autonomic nervous system, this is something that we'll be covering coming up, but when you see that word autonomic, think automatic. Smooth muscle generally is, the cells themselves are rather fusiform, and they generally have a single nucleus. My pen will cooperate here. I don't know what's going on with that. Okay. So generally fusiform cell, single nucleus. We don't see striations. The sarcoplasmic reticulum is not as abundant as it is in skeletal muscle tissue, and you don't see T-tubules. And this has to do with just how calcium is utilized within these cells. And then we do see thick and thin filaments, but we don't see striations. So there are a couple of categories of smooth muscle tissue that relates to how they are innervated. So innervation with smooth muscle is typically a little bit different than what we saw with skeletal muscle. So recall that in skeletal muscle we had this fairly precise junction between the axon terminal of a nerve and the muscle itself, known as the neuromuscular junction. With smooth muscle, there's generally not going to be precise neuromuscular junctions. And as a result, we have these two different categories of smooth muscle. We have what's known as multi-unit smooth muscle. This is typical of large muscles as well as pulmonary air passages, the piloerector muscles, and then muscles in the eye that control how relaxed or dilated the pupil is. And then the innervation in this case is going to be similar to that of skeletal muscle, though in smooth it's autonomic instead of being from somatic motor neurons. And then with single unit, this is where it really differs from skeletal muscle. With single unit smooth muscle, this is going to be typical of the walls of blood vessels, digestive, respiratory, urinary, and reproductive tracts, so it's a little bit more common. And with this it's single unit because it's multiple cells contracting as a unit. So even though you have multiple individual cells, it's like they're all working together. And in this case, the myocytes are electrically coupled to one another. And we have these swellings within the autonomic nerves that are known as varicosities. The varicosities are going to be releasing neurotransmitter, and that is going to basically stimulate the cells in mass rather than at these precise neuromuscular junctions. Okay, so that is going to wrap up my lecture that covers Chapter 11 of the Saladin text. As always, please let me know if you have any questions. And like I said, I would encourage you to read the parts of the chapter that I didn't get to cover in lecture just due to time limitations. Again, especially if you are looking at physical therapy, occupational therapy, or exercise and sports science, because a lot of these concepts will come up repeatedly in further coursework.